Welcome to another edition of CCSD Living Environment. Today what we're going to look at is going from the language of nucleotides to the language of amino acids. So like we said last time, how do we go from this code in the DNA, these letters A, C, G, and T, to actually getting a human being, to actually getting an organism? How do we do that? How do we go from that message, that code, to actually getting a person? So we've got a couple things that we need to review before we can go into too much detail. First of all, proteins are large, complex molecules, remember polymers, made of smaller repeating subunits or monomers called amino acids. That's the first thing we need to talk about. Then we should define the word gene. A gene is a section of DNA that codes for a protein. So therefore it determines the trait. The traits that you have, eye color, hair color, skin color, height, things like that, all of those things are based on proteins. And those proteins come from the DNA, a specific sequence of DNA called a gene. And that takes us to a new word called phenotype. The phenotype is the observable or physical trait that an organism has. Like, for example, hair color, eye color, the different things that we see when we look at a person or some other organism. And then the opposite, I guess, if you will, of that is a genotype. The genotype is all of the genes or the genetic makeup of an individual. So the phenotype is what you see when you look at somebody. It's what's actually been expressed. And then the genotype is the actual gene itself. It's what is being expressed. So we need to have a little bit of understanding of those terms for us to proceed further. And we need to come back to RNA. We need to revisit RNA for a minute. Remember, RNA contains 4 bases. A for adenine, C for cytosine, G for guanine, and U for uracil. And let's just throw out there one more time, RNA is made of nucleotides. Remember, the RNA nucleotide has ribose for a sugar instead of deoxyribose, and it has U as a base instead of T. So RNA is very similar to DNA, but there are some pretty big differences between them. Also remember, DNA is double-stranded. We have a double helix. RNA is single-stranded. It's just basically a chain of nucleotides hooked together. So based on all of that information, how do we go from that nucleotide language, these letters, to actually amino acids or a protein? The bases in RNA form a language using just those letters A, C, G, and U. This language is called the genetic code. It is read 3 letters at a time so that each word is 3 bases long and corresponds to an amino acid. So we know that in our language there's a whole bunch of different combinations of letters that come together to make different words. Well, in the language of nucleotides, in the language of genetics, every word is only 3 bases or 3 letters long. And those 3 base long words are called codons. So each 3-letter word is called a codon. And each word corresponds to a specific amino acid. Now, if you remember from our last unit, we said that there are 20 different amino acids. So that means that there's got to be at least 20 different combinations to give us each amino acid. To decode the codons, a genetic code table is used. If you look in your note packet, you will see there's a little chart there with a circle, and it shows you how to go through and take like ACC and give you an amino acid. So the same way you decoded things when you were a little kid in puzzle books and stuff, we're going to use this language of nucleotides and decode it using the genetic code to determine what the amino acid sequence for the protein is. So we're using the mRNA to do this. The same way that our language has punctuation, we have the beginning of a sentence, we have the end of a sentence, the genetic code also has that. We have a start codon, which we're going to call AUG, and there are 3 stop codons, UAA, UAG, and UGA. And right there in your note packet, it has a little practice thing for you to do, to practice using the genetic code, to go from the DNA, which notice has T's in it. You're going to make the mRNA. Remember, it is complementary, but this time you're going to put U's instead of T's. And then there's a couple other questions there for you to answer. And we will come to those tomorrow when you bring that information to class. So once we go through and we do that, we need to actually look at protein synthesis. Protein synthesis is the process where a protein is made from an amino acid. So the protein, remember, is the polymer. That is the very large molecule made from the same repeating subunit, the monomer, in amino acid. So if I put amino acids together, I get a protein. If I break down a protein, I get out amino acids. The process that translates an organism's genotype into its phenotype. I'm going from the genes, I'm going from the A's, C's, G's, and T's in DNA to the phenotype, the actual amino acids that are in the protein. And what's the thing in the middle that gets me from the DNA to the protein? That's where the mRNA comes in. The sequence of DNA in a gene acts as a template or pattern to determine the sequence of amino acids that make up a protein. A gene is expressed when the protein it codes for is synthesized or made. So why do we have to have this other step? Why do we need the mRNA? Well, think about our cells. The DNA is in the nucleus, the ribosome is out in the cytoplasm, or it's attached to the endoplasmic reticulum. Something needs to get that message, something needs to get those instructions from the nucleus to the ribosome. And it's the messenger RNA that's going to carry that information for us. So let's keep looking at protein synthesis. The ribosomes use the sequence of codons, remember those were the 3 letters in mRNA, to assemble amino acids into a polypeptide chain. Remember, a polypeptide is a protein. Polypeptide because when you put many amino acids together, there are many peptide bonds. The decoding of an mRNA message in a protein is called translation. So let's review for a minute. When I went from the language of DNA, or nucleotides, to the language of RNA, or nucleotides, we called that transcription. We're within the same language. But when I go from the language of mRNA, nucleotides, to the language of proteins, amino acids, now I'm changing languages. It's like going from English to Spanish. When you change languages, you need to translate the information. So transcription was DNA to RNA. Translation is RNA to protein. I'm going from the language of nucleotides to the language of amino acids. Different language, so I have to translate the information. So what are the steps to translation? First of all, the ribosome will attach to the mRNA in the cytoplasm. As the mRNA code enters into the ribosome, so think about it as a long chain and it enters into the ribosome, kind of like a roller coaster coming into where you get off. A tRNA molecule, a transfer RNA molecule, will bring the correct amino acid to the ribosome. So the tRNAs are out floating all over the place in the cytoplasm. And when that mRNA, when that roller coaster comes back in and it starts to come into the ribosome, tRNA comes down. Each tRNA molecule can carry a specific amino acid. Well, how does the tRNA know, first of all, which amino acid am I supposed to carry? And second of all, how does it know that it's matching up the correct amino acid to the right codon in the mRNA? That's because tRNA carries the anticodon. The anticodon is complementary to the codon. So if I am mRNA and my codon reads AAA, then as tRNA, my complementary anticodon would read UUU. You're right. It's the complement, and we're in RNA, so we have to have U, not T. So it's kind of cool because we know that tRNA is going to carry those amino acids in. That's how the amino acids get to the ribosome. So the mRNA has the code that it's bringing from DNA, and tRNA is saying, okay, well, I have to know which amino acid to carry, and I have to make sure I bring the right one in at the right time so we get the right protein. So how does it do that? Well, to get that correct protein, what it does, it has an anticodon that is the complement to the codon. So it brings in that amino acid. All right, what happens next? The ribosome helps form peptide bonds between the amino acids. Once the bond forms, the tRNA lets go of the amino acid and he goes to get another one. So think about this process. Here's my ribosome. The mRNA has come in like a roller coaster coming in, and it comes into that ribosome. And so, the brakes hit, whatever, however you want to think about that. And those first 3 letters are there. So tRNA from the cytoplasm comes in with this amino acid, codon, anticodon, complementary matchup. And so now we kind of shift down a little bit and we check that next codon. The next tRNA comes in, the 2 amino acids hook together by a peptide bond. Once that peptide bond is there, this tRNA doesn't need to be there anymore. So he lets go of his amino acid and he goes back out into the cytoplasm. Well, what does he go do? He goes to try to find another amino acid. Can he pick up any amino acid he wants to? No, he has to pick up that same specific amino acid he just had. And then if that codon happens to pop up again, that anticodon will match up and he'll bring back in that amino acid. And they just keep doing that. And it goes all the way down through. And when does it finally decide, okay, this process of making the protein is done? When it hits that stop codon. Remember we said there were 3 stop codons. So it'll just keep going and pulling all those through and bringing in the tRNAs with the amino acids until it hits a stop codon. When it hits the stop codon, the ribosome lets go of the mRNA and the protein goes off to go do whatever it's going to do, whatever that protein is going to be. Whether it's going to be an enzyme or it's going to be an antibody, it's going to be a hormone, a component of your cell membrane, whatever that protein is going to be, it's going to go do its job once it's finally made. So the polypeptide chain or protein grows until the ribosome reaches a stop codon on the mRNA. This is the end of that translation process. This is the end of going from the language of nucleotides to the language of amino acids. So now that we've kind of talked about what's happening in the ribosome, and if you look there in your note packet, there's a couple pictures. There's a picture that shows you the cell, and it shows you the nucleus inside of the cell. And it goes through to show you that the DNA is turned to RNA in the nucleus. The RNA leaves the nucleus to find a ribosome. And the tRNAs bring the amino acids in for protein synthesis to take place. The bottom picture there just shows you the tRNA coming in, the peptide bond being formed between the amino acids, and then the tRNA letting go of that amino acid and going off to find another of the same one. So this is kind of summing up everything we've talked about. The DNA is going to be transcribed to RNA, and the RNA is going to be translated to a protein. So I'm going to go from the language of nucleotides to the language of nucleotides, transcription, to the language of amino acids, translation. When I change languages, that's when I go to translation. So here we're talking about the genotype. This is the genetic makeup. This is the part we can't see. So I'm going from genotype to phenotype. This is when your genes are expressed. This is actually what we see when we look at the traits or the characteristics of a person or any organism. So we're going from genotype, those A's, C's, G's, and T's, to the phenotype, your hair color, your eye color, how tall you are. Any of those traits that you physically express that we can see when we look at each other. So we're going from genotype to phenotype. We're going from the language of nucleotides to the language of amino acids. That gives you a little bit of an idea for how we go through and actually go from A, C, G, and T to me. So it gives you some idea for how that takes place. Please make sure you take a minute to look at the pictures. They're very well laid out in the note packet for you. They give you lots of good information. Everything is very clearly labeled. And the next time when we come back, what we're going to look at is what they call the central dogma of genetics. Have a great day. I'll see you later.